Hello and welcome to the next review video. This one is about B22, organelles and compartmentalization. So, what are organelles? Organelles are discrete subunits of a cell with specific functions. So they have some things they want you to know are organelles, like nuclei, ribosomes, vesicles, plasma membrane. These all count as organelles according to IB, and there's more that we'll talk about later. That's just standard number one. But there are some that they don't count, and that is cell wall does not count, cytoskeleton does not count as an organelle, and cytoplasm. So all these kind of like structural things do not count as organelles. They don't have a function, they're just structural. I think that's their reasoning. Nobody really knows. How did we discover these? Freeze fracturing was a method of describing them all. Ultracentrifuges and freeze fracturing. Great. 2. Nucleus versus... Why do we have a nucleus? What is the advantage? 1, it protects the DNA. Like there's more barriers, so it protects the DNA. 2, it allows for post-transcriptional modification. So what that means is we have DNA to mRNA, and then mRNA to protein outside the nucleus. And so since there's this gap, you can do things to it. And so those, we'll learn more about these later, but it's going to be like our exons, our poly A tail, and our G cap. And so there are 3 things that can be done to it before you export it. And so it allows you to do these post-transcriptional modifications. In prokaryotes, it's not possible because there's no barrier to stop this. So this is done by ribosomes. And those ribosomes can just get all the way right here. And so you don't have this time where you can do post-transcriptional modifications. 3. What is the advantage of compartmentalization in general? So these are our eukaryotes, right? Have compartmentalization. And so it allows you to concentrate metabolites in one spot with the enzymes. Concentrate metabolites and enzymes together. So for cell respiration, right, you can put them all in the same spot, and then yay, it'll make the reaction happen faster. So this one is all about speeding things up. And the other thing is it separates processes. And this is all about protecting other parts of the cells. So you have typically in animal cells, we have lysosomes, and then we can also have phagocytic vacuoles. These things are both all about destruction. And so they're all about breaking things apart. And so they have a bunch of digestive enzymes. But they're locked in a membrane, right? So they're membrane bound so that they can't just go around destroying your entire cell. So if you have these digestive enzymes like chop things up, you wouldn't want those floating around your cell. That would be really bad. And so that's separating the process. Lysozymes and phagocytic vacuoles. Vacuoles are a good example of that. because they separate the digestive enzymes and prevent it from destroying things in the cell. They also have a single membrane, because they're basically just vesicles. I have seen that as an IB question before. That is the end of SL, so now we're on to the HL only section. Mitochondria. So the mitochondria has a double membrane, which creates the intermembrane space. The intermembrane, so the double membrane is important because it allows for the creation of the intermembrane space, which has a small volume, which means you can get a high hydrogen gradient, because your gradient is the number of particles over the volume. So if you have a small volume, you can get a much higher proton gradient, and so that'll help with the ATP synthase. You have a large surface area of the cristae. So a large surface area means more ETC, and so you can embed more proteins into the membrane because you have a larger membrane. And then the matrix is that concept we talked about in the last part about putting all of the reactants and enzymes together. And what process happens in the matrix? Krebs cycle. Way to go, you got it. So proud. Nice, that was number 4. Good work, team. 5, chloroplast. Gonna be very similar, right? Just different names, but same concept. So, thylakoid membrane has all those discs, and that's gonna be surface area as well. So it increases the surface area. So the thylakoid membrane has the discs, so it increases the surface area. So that means more photosystems. So that means you can absorb more light. So you have more pigments. Oh, so good. The thylakoids, right? Inside those discs, it's a small volume, which means you can have a high hydrogen gradient, concentration gradient. This is important because it doesn't actually change the number of hydrogens, it just changes the gradient. And then, of course, stroma is for this Calvin cycle reactants and enzymes. Oh, I could have made it a question. Sorry. Quick pop quiz. What's the main enzyme you need to know for the Calvin cycle? 3, 2, 1. Rubisco! Nicely done. Alright, that's the chloroplast. 6. Back to the nucleus again. Nucleus. So it has a double membrane with pores. Pores allow for mRNA to leave to the rough ER. They also allow it to just leave in general. That double membrane provides extra protection, and because it's got this membrane, it can break into vesicles during mitosis and meiosis. And so what you do is you never actually destroy your nuclear membrane. You put it into vesicles, and then it can reform. So it can reform from those vesicles, because remember, vesicles are just like this, and then if you push them together, they just go like that, and they become a bigger one. And so the main thing, right, is the pores allow the mRNA to leave, and then because it's got this double membrane, it allows it to form vesicles, and the vesicles can be temporary, and then you can smush them back together to reform the nucleus at the end of mitosis and meiosis. All right. 7. So you have free ribosomes and rough ER ribosomes. These are proteins for the cell. And these are proteins for the membrane or secretion. So those are ones that are either going to be membrane bound or secreted out, not kept inside the cell. So that could be like hormones or enzymes, right, that you're sending outside the cell. Okay. All right, 8. Golgi apparatus. The Golgi apparatus is used for processing and secretion of protein. It actually has other functions, but that's all you need to know. So what is the structure and function of it, right? So basically, it looks like this, and what it can do is it can bud off, like, it can go like this, and then it can go like this, and then it can go like, woo! And so inside of here would be your protein, and so then it creates vesicles, basically. So it creates vesicles filled with protein. Awesome. Good work, team. 9. Oh, vesicles. Ooh, so vesicles, right, are for moving things inside of cells or out of cells. They're single layers of membrane bound together with stuff in the middle. They're formed by clathrin. So clathrin is that molecule that would cause this process to happen. And so basically it's like this, and clathrin is like, hey, 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 fold over. Fold over now. And so you start to get more and more clathrin around it, the more it folds over. And the clathrin is like, yeah, make it, and then eventually it becomes a vesicle. So clathrin stimulates it and causes it to fold inward and create that circular vesicle that we know. So clathrin is the protein that is responsible for vesicle formation. And that is B22, organelles and compartmentalization.